Yay. All right. Now it's working. All right. Really quick for attendance. Let's see here. I see we have Boyd and White have joined us online. Good morning to you too. All right, 7:59. I'll go ahead and get started with announcements. Save us a little bit of time with the actual lecture. All right. Really quickly for attendance, I'll remind you guys, obviously, for those of you online, send me the email with the keywords. But from now on, just make sure you get it in by 9. I have reasons. I don't want to get into it. I'll answer your questions if you want to know why. If you meet me during office hours, I can explain why. But just know I want you to get the keywords in by 9. Also, speaking of attendance, well, we've got one person here, so you're good to go. Obviously, you don't need to do anything different. I've already got you down. For anybody else or on a different subject, anybody who has absences, old absences, you can look at that grade spreadsheet to see what days I have you as absent or late, and you can go back and watch old videos for that. So if you were late, you can watch the video, answer the questions, and bump your grade back up to full score. If you were completely absent, you can watch the old videos and get half a score, but that's better than 0. So any questions about attendance? All right, lab. I know I put this out there yesterday in writing, but I want to put it, say it verbally too. Make sure you're on time for lab, because usually labs are some sort of team effort, right? Even if you're working alone, usually somebody else needs your data. So we need you there on time, because the rest of everybody else is going to be relying on you to be there. So make sure you're on time. And I know I put this in writing yesterday, but if you're not there at all, you're only going to get a quarter credit for your lab, unless it's excused. If you've got COVID and you're not allowed on campus, there's nothing I can do about that. So you'll get full credit for that. But if you just don't show up and yet you do the lab, you'll get 1 quarter credit. If you show up late and or leave early, then you'll get half credit. So be there on time. Be ready to go. That's what the pre-labs are for, to get you ready. Because again, sometimes labs can take a long time, and I know people don't want to sit around a long time. So be there ready to go. It'll save you time. What else? Really quick reminder, and you'll get this reminder again in writing today. The lab 4 is due tonight. So what was lab 4? That's the Osmosis Diffusion lab. Make sure you have that done by tonight. The pre-lab is due by Sunday. The lab 5 pre-lab, which is the photosynthesis respiration, make sure you have that done by Sunday. As always, the earlier you have it done, the better. Hopefully today I can go through all of them and see what you've already done and start giving you some feedback. So hopefully you can fix it before the due date and get full credit for fixing it. Any questions about lab? All right. I'll remind everybody, I've said this before too, but recently this is a new change for office hours. Any lab you've turned in or any exam, lab or exam, if you come to me during office hours or schedule me something, sometime after office hours, and allow me to sit with you and give you feedback, like for the exam, for example, I can go through and tell you why your wrong answers are incorrect. If you allow me to do that, I'll give you a 5% boost on any grade, whether it's a lab or an exam. If you let me torture you with feedback, then I will boost your grade, because really I'm here to teach you, right? So if you let me give you feedback, that's going to help you learn. So I want to, give you incentive to do that. Any questions about that? All right. The last reminder, independent work, please get to it. See me during office hours if you really still don't understand it. Basically, you need to write papers, but you need to write papers. Write your own papers. This is the last time I'm going to say this. The best thing you can do is read an article that you're interested in, read it however many times you need to, and then put it away. And then from your memory, write what you remember. Because you're not going to lose points if you don't remember a lot. You can literally write a paper and say, I was wondering why pine cones hurt so much. And I read a paper, and the scientist, I don't remember the scientist's name, but they said that they're spiky because it had something to do with evolution. You can be vague. That's okay. Being vague is better than rewriting something or copying something. So make sure you do that. And anyway, independent work. See me during office hours if you need help. I can give you some help. There's other things you can do. I can give you some inspiration. Any questions about any of the announcements? All right, let's move on to it then. Let's get back into chapter 5. Here we go. To remind you, last thing we were talking about was enzymes. We talked about the fact that they're proteins. That's an important thing you need to know. We talked about the fact that they are not used up in chemical reactions. So when you see a test question and I'm saying which one of these things is the enzyme, if it's on both sides of the equation, that means it's an enzyme, right? Because it wasn't used up in the reaction. It's not necessarily a product or a reactant. So that's one way of knowing it. Another thing we said that you need to know is that as far as we're concerned in this class, enzymes end with the suffix ase, A-S-E. So again, if you get a test question, I say, hey, what's the enzyme? And you've never seen those words before. If it ends with an ase, that is the answer. Another thing we talked about that you need to know about enzymes is they are generally named after their substrate. So for example, lactase is the enzyme that breaks down the molecule lactose. So again, you'll get a test question with some chemical equation you've never seen. And one of the questions might say something like, what is the enzyme? And look for the thing that's on both sides of the equation that has the suffix ase. And then you'll get a question that says, what is the substrate? And once you figured out the enzyme, just look for the thing that has a similar word to the enzyme that doesn't end with ase. So again, lactase being the example. Lactase is the enzyme. Lactose is the substrate. We also talked about how enzymes, the way they work is basically they lower activation energy. And I use that analogy of pushing a rock over a hill. You know, that rock has a lot of potential energy. And all you need to do is get it rolling. And once it gets rolling, it'll roll down the hill, right? All that potential energy will be released and it'll be turned into kinetic energy. And the same thing happens with chemical reactions. Usually there's energy released during a chemical reaction, but you still have to push a little energy into it to get it started. And that's what enzymes do. They lower the amount of energy you need to get it started. So that's when we're now caught up with enzymes. Really quickly to remind you what we talked about up there, ATP. Basically, I mean, there's a lot you need to know, but I'm not going to go through it all again. The most important thing you need to know from that bullet point right here is that cells need ATP for energy. Some people got that wrong. I don't remember if it was the study guide or the attendance questions, but yes, cells need ATP for energy. That's, if anything, that's the most important thing you need to know from this chapter. And of course, a long time ago when we first started the chapter, we talked about some basic energy concepts, potential energy, kinetic energy, conservation of energy, all that stuff that I'm not going to rehash, but you can see me if you need help with it. So anyway, that catches us up. Let's talk about enzyme inhibitors. First of all, does anybody know what an inhibitor is? Forget the fact that we're in a biology class and think about the word inhibitor or inhibits or inhibitions, any version of the word inhibitor. Does anybody know what that word means? No guesses? A process? That's good. That's really good when you're thinking about biology, but think about outside of biology. Like, what if I said, you know, my friend's a real introvert, but when we go out drinking, you know, he gets a few beers in him and all of a sudden his inhibitions are lowered. What am I talking about? What are his inhibitions? What is that? No guesses?
It doesn't have to do with like you say your friend. I'm using your example like if you're playing this is the right one. And like his
Energy? That makes sense too. All right. I like you guys are thinking right along the right tracks, especially if we're talking about enzymes. I'm so glad you said energy. Even though it doesn't necessarily have to do with energy, at least you're thinking about enzymes, and enzymes are all about energy. So inhibitions, using the same example, you know, if you say if I'm drinking, it's lowering my inhibitions. That means inhibitions are things that are keeping you from doing something. Like inhibitions are keeping you from talking to people, or inhibitions are keeping you from acting a fool, or inhibitions may be keeping you from starting a fight or hitting on somebody or whatever it is. Inhibitions keep you from doing something. So keeping that in mind, an inhibitor is something that keeps you from doing something, specifically an enzyme inhibitor. And this is what you need to know before I even get into this bullet point. If anything, just know this. An enzyme inhibitor is just something that keeps an enzyme from doing what it's supposed to do. I'm going to give you some more details. I'm going to tell you how those work. I'm going to give you some different categories of it. But if anything, just remember that enzyme inhibitors, they just keep enzymes from doing what they're supposed to do. So here we go. Enzyme inhibitors, the way your book says what I just said is they inhibit metabolic reactions. Because remember, when we first started talking about enzymes, I introduced you to the word metabolism. I said metabolism is all the chemical reactions that are happening in your body. And then I went on to say nearly every single one of those chemical reactions wouldn't even happen without an enzyme. So here we're back to that concept. So yeah, enzyme inhibitors inhibit metabolic reaction. That's the book way of saying enzyme inhibitors keep enzymes from doing what they're supposed to do. How does that work? How do they do that? Well, they bind to an enzyme and they disrupt the function. I'm probably not going to ask you those two specific things. Like for the exam, you don't necessarily need to know that they bind to the enzyme and they disrupt the function. I'm about to talk about those two things a little bit more in detail. So right now, those two bullet points are more of a preview. But again, so far, the most important thing you need to know is that enzyme inhibitors just keep enzymes from doing what they're supposed to do. So there's different types. The first one we're going to talk about, number one here, they're called substrate imposters. And what do? They plug up the active site. So this is a good time to remind you some of the things we talked about at the very end of the last lecture. And I'm going to use this picture even though it's a meme. Let me hide this. There we go. It's still a pretty decent picture. So here, this yellow thing, that's the enzyme. I'm going to put an E for enzyme. This right here, that's the molecule that the enzyme does its job on, right? So it's the S. I'm going to put S for substrate. And then this part right here that's shaped like the substrate, that's where the substrate fits in. And that is called the active site. I'm going to put AS as the abbreviation, right? So to remind you, that's how it works, right? The substrate comes into the enzyme at the active site. Then you get that induced fit where the enzyme kind of wraps it around a little bit tighter. That's how enzymes work. Now, that in mind, now that I've reminded you of that, the imposter, one of the types, the first one we're talking about, the substrate imposter, it just goes into the active site. It plugs it up. And even though, again, this is a meme that I didn't make, I like it because it really does show what it, shows what happens. You can see that there's the enzyme, here's the substrate saying, hey, that's my enzyme, which is cool, because the enzyme is being, the substrate's being very possessive. That's my enzyme, which is I know it's only slightly funny, but I like it because it's pretty accurate, because this is a good time to remind you that, remember, enzymes are very specific about their substrates. And they catalyze very specific reactions. So anyway, then you got this other thing. It's obviously not the same thing as the substrate. It looks different. It's a different color. It's a different size, but it happens to have at least part of the same shape where it could go in and plug up the enzyme. And that's how it works. It really just plugs it up and then it doesn't work. I'm trying to think of some real life examples. Slightly. All right. I'm sure you guys have seen those little plastic things that you put in wall outlets when you have kids and babies around. Yeah, the outlet blockers. Yeah, that's sort of the same thing, right? Like you put those in there and you're doing it for safety. But the reason is you're keeping those blockers from doing their job, which is putting metal in there and it conducting electricity, right? Or, the red spots at WVSU are supposed to be for faculty. And if you were to park there and you're not student or faculty or whatever, you're blocking somebody, right? So that if you consider the parking spot the active site, you're keeping somebody from parking there. And the job of the parking spot is to let somebody, a faculty member or a student, depending on where it is, park. So yeah, it's pretty simple, right? It just plugs it up and it keeps it from doing its job. Any questions about this first one, which is substrate imposters? All right, let's talk about the other category. Number 2, and there's a name for it, but your book doesn't mention it, so I won't either. Because that just means I'm not gonna ask you since I'm not telling you what it is. You can look it up for independent work if you want. But the first one we talked about that we just got finished talking about, that was a substrate imposter. The other one, I'm not even giving you the name of it. I'm just gonna tell you what it does. It binds at a site remote from the active site. In other words, the last thing we talked about literally plugged up the active site, right? That's where it binds. This one binds somewhere else. So it's a good time to bring back what was on the other side to remind you. Whatever kind of inhibitor we're talking about now, even though I've said it, now you've seen it. No matter what kind of inhibitor we're talking about, it binds to the enzyme. It might be the active site. It might be somewhere else. Either way, it binds to the enzyme. So that part, obviously, you need to know. And again, this second category that we're talking about, it doesn't bind in the active site. It doesn't block the active site. It binds somewhere else. So you might be wondering, how does that do anything? Well, this is how it happens right here. When it binds, it changes the enzyme's shape. And this is a good picture of that. So here's the, here's this enzyme. You can see the substrates kind of look like a, I don't know, like pumpkin teeth, right? It's got that shape to it. And you can see that it fits in the enzyme perfectly. Well, what happens is, see, this is not from your book, and I gave it away. This is called allosteric site, but you don't need to know that. So I'll put a line through it. So this other site, other than the active site, the enzyme comes in, it binds to it, and look, you can see here on this right-hand side that the enzyme, the active site has changed shape now. So it went from square to triangle, right? All because this inhibitor binded to it somewhere else. And that's how it works. That's how the other type of enzyme inhibitors work. They bind somewhere other than the active site, but that changes the shape of the active site. And again, that keeps it from doing its job. Yeah, it's pretty simple. There's not much else to say from that. Obviously, this is a good time to remind you of all the key things that you need to know about enzymes. Their shape, it's all about the shape, right? Just like any protein. It's all about the shape. Its function is based on the shape. So if you mess up the shape, which is what this does, then it doesn't do its job. Any questions about that? Okay, let's move forward. Here's some pictures of all three things that we're talking about. This is actually from your book. This first picture up here, letter A, this is just things working as normal. That's an enzyme and its substrate. There's the enzyme. It's got a nice little circular active site. The substrate fits in there perfectly. Yay, things are working pretty nicely here. B, this is the first one we talked about. The enzyme is inhibited by a substrate imposter. So here you go. You can see this thing right here, this inhibitor. It's not the same exact shape as the substrate, but it's close enough. It's close enough to where it can fake the funk and come in here to the active site of the enzyme and block it. So now because of that, the substrate can't come in. It just bounces off. And again, it keeps it from doing its job. And then the last one we talked about right here, letter C. Again, this thing binds to the enzyme just like the other one, except it binds for the different spot. It doesn't bind in the active site. It binds somewhere else. In this case, it just changes the shape of the active site. So now the substrate can't fit. And again, it bounces off. It keeps the enzyme from doing its job. So all that to say, enzyme inhibitors keep enzymes from doing their job. And there's basically, and no matter which kind it is, the way they do it is they do it by binding to the enzyme. There's two different versions. One of them binds to the active site. One of them binds somewhere else. And that's it in a nutshell. Any questions about that? All right. This is pretty simple stuff, I hope. All right. This is kind of important, not for the exam, just for life in general, just to understand how these things work. The binding of the inhibitor can be reversible. I'd have to look this up, but I would think, if I remember correctly, and maybe I shouldn't have even emphasized the word can like that, like can be reversible. I think most of them are reversible. Because think about it. Again, metabolism is all the chemical reactions happening in your body, and you wouldn't be alive without those chemical reactions. And almost every single one of those chemical reactions happens because of an enzyme, right? So you wouldn't want these things to be permanent. So let's, for now, I'm going to go ahead and cross through that. Yeah, they can be reversible. I just don't like the way I emphasize the word can. I'll abbreviate here, UA. And again, I don't know. You might look this up. Usually are reversible. I don't remember. That's not important. The important thing here is that know that it's possible for them to be reversed. And that's not a trivial piece of information, because sometimes that is very necessary. Sometimes you need to turn an enzyme off, but then later on you need to turn it back on. Because remember, an enzyme is just making a certain chemical reaction happen. And sometimes chemical reactions, for example, are building something, right? Maybe you need a certain molecule. So does anybody remember those fidget spinners? The kids, they were like the big craze not that long ago. And I don't know, you guys might be too old or too young, except maybe one of you. There was also a time a while ago, like even when I was really young, there was a Beanie Baby craze. Like, I don't know, people spent hundreds, thousands of dollars on these little stuffed animal things. Anyway, there's crazes, just like the fidget spinner. My point here is, imagine if you owned a toy factory and you could build all these different toys, but then like the fidget spinner comes up. You're like, all right, everybody stop what you're doing. Let's put all of our resources into making those fidget spinners, right? No one's buying this other stuff right now. Let's just spend all of our resources and time and effort into building these fidget spinners. That's a good thing at the time because they were selling like hotcakes. But then once the craze went away, then you need to be able to say, okay, it's not time. Let's not build so many fidget spinners anymore, right? Let's start building these other things, right? And you put your resources and your workers back into building other stuff. It's the same thing with your enzymes. Your bodies need molecules. So these enzymes are like, I got you. And then they're making the enzymes, or excuse me, the enzymes are making these molecules that your cells need, that your body needs. And then once they have enough, something's got to say, all right, quit pouring resources into making those molecules. We have more than we need. You're wasting material. You're wasting energy making these molecules. We don't need any more. And that is what enzyme inhibitors do. I mean, you know they stop enzymes, but now you know why they do that. Why would they do that? That's why. Because sometimes your cells just need to stop making a certain molecule, whether it be permanent or, like we're talking about here, reversible. So yeah, again, using the fidget spinner thing. So you have your factory. Fidget spinners are no longer a big craze anymore. So you go back to making other toys, and you're still making a few fidget spinners. But then maybe a year later, they become a craze again. So then you do the same thing, right? Then you start pumping up and making more fidget spinners. But again, the idea here is that you turn it on, you turn it off. Same thing with enzymes. Anyway, any questions about that? Because again, sorry, I just want to point this out because this little bullet point right here reminded me, that last bullet point. It keeps cells from wasting resources. And we don't really think, a lot of us don't think about that very often, but energy is not infinite, and neither is material, right? So when you're on your body, when your cells are building things, you don't want to be wasteful. You don't want to use energy because you need energy. You have to get energy, right? And material, you have to eat. So anyway, that's why it's important. Any questions about enzyme inhibitors being reversible? All right, this next slide, if I remember correctly, gives you some examples of drugs that do this. And this is just an example. This is not a pharmacology class, so you don't need to know this for the exam, but here we go. Many drugs work by inhibiting enzymes. Penicillin is a good example. That is, as you probably know, an antibiotic. So if you have a bacterial infection, it kills the bacteria. How? It blocks the active site the bacteria use making cell walls. So again, remember, like I said, enzymes, there's like an enzyme for nearly everything that a cell does. So in this case, bacteria have something called cell walls, and that protects them. So what happens is penicillin goes in there, it binds to the enzyme that's making the cell wall. So now it's no longer making the cell wall. Therefore, the bacteria has, I don't want to say no defense, but as far as this conversation is concerned, sure, it has no defense. And then your body's able to kill the bacteria. So that's one example, penicillin. Ibuprofen, if you're a veteran, I don't remember if any of you are, but that's a big thing for veterans, like in the military. They give you that for everything. You get shot in the leg. Like, here's 800 milligrams of Motrin. Take that and drink some water and you'll be fine. Anyway, ibuprofen, that is, it inhibits the enzyme that sends pain signals, right? So even your neurological system, that thing is sending messages to and from your brain, there's enzymes responsible for that. And one of them can be inhibited with ibuprofen. Cancer drugs, a lot of cancer drugs, they inhibit enzymes that promote cell division. And you're going to learn all about this for the third exam. Basically, as far as we're concerned in this class, cancer is just cell division gone crazy. So if you can control the cell division, then you can control the cancer in theory. So that's how a lot of cancer drugs work. Anyway, and then finally, your book also points out that many toxins and poisons also work as inhibitors. And that goes way back to the very first slide of chapter 5, when there's a person in a yellow hazmat. And if I remember correctly, they had their arms crossed like this. They had a yellow hazmat suit and a gas mask. And that's what your book was getting at. And they said that's how nerve gas works. I forgot specifically which enzyme it is, but that's how it works. It goes in there, it binds to a certain enzyme, and it basically stops your nervous system, and that's what kills you. Anyway, again, this whole slide, not on the exam. This is just saying, hey, here's some practical stuff that you might know about. So any questions about that? All right. I haven't even given the first word for attendance yet. I'm sorry. The first word then will be, yeah, there you go. Yeah, we'll do that instead. I'm taking suggestions. The first word is Motrin. I'm circling it right there, Motrin. Not ibuprofen, which is the generic name, but Motrin. All right. Here we go. Now you've got your first word. So that's it. We're done talking about enzymes. Now we're going to talk about membrane function. And I wish we were a little bit ahead, because this is the kind of stuff that you should have known before you went into lab. Then again, now that you've had it in lab, maybe hopefully it'll be a slight review for you now. So let's talk about membrane function. Before I even get into this, I posted 2 videos yesterday. Whoops. Oh, what happened here? Anyway, I'm going to post more videos like the ones I posted yesterday. And I really highly, highly recommend watching them, because one of the things they're going to talk about are membrane functions, which is what we're about to talk about. And before I even get into this, I would just want to make sure you understand why this is even important, right? Because otherwise you'll just be sitting here bored and be like, why am I bored? Why does this matter? Remember, cells control their environment. That's one of those properties of life, right? So they have to be able to control what comes in and what comes out. Without that, we would not have life. You wouldn't exist. I wouldn't exist. There would be no life on Earth without cells being able to control what comes in and out. And one of the big ways they do that is this bullet point right here, membrane function. So everything we're about to learn is the basics about how cells control what comes in and out. And again, without this, as boring as it might seem, and as easy as it's going to seem, I hope, it's very important, and we would not have life without it. So here we go, membrane function. That first bullet point is pretty much what I already said. Cells regulate the flow of materials to and from the environment, right? Cells control what's inside them. They're generally not, what's inside the cell is generally not what's outside the cell. Hopefully you saw that with your experiments, right? You had a little cell that had starch and salt, or maybe it had sucrose. And hopefully you saw some things moved, some things left, some things came in. Anyway, how does this happen? Well, there's something called the plasma membrane. And you do need to know this. The plasma membrane consists of a double layer of fat, which is what we call a phospholipid bilayer, that is embedded with proteins. That's a lot of information. For right now in the semester, the only thing I want you to know, I mean, you can know all that other stuff too, but the most important, if you forget everything else, just know that the outside of your cell is called the plasma membrane. Just know that. The rest of it, yeah, you can know that if you want, and we'll come back to that a little bit later. But for now, just know that that's what the outside of your cell is made of. And remember, we skipped chapter 4. And had you read chapter 4 and had we covered it, you would have learned a lot more about the cell membrane. But again, the cell membrane, that's what's on the outside of your cell. So basically, a cell wouldn't exist without the cell membrane. Otherwise, it would just be a bunch of stuff out in the middle of nowhere, right? So the cell membrane kind of defines what the cell is. That is not to be confused with, I'm going to point this out very quickly, because there's a question, I forget where, maybe on the study guide, maybe on my, maybe the lab, maybe my attendance questions. Anyway, the cell membrane is different than a cell wall. So for example, we just got finished talking about penicillin and a cell wall for bacteria, right? Bacteria, some of them have a cell wall, but they also have a cell membrane. Every single cell on Earth has a cell membrane. That's part of what makes a cell a cell. They have a cell membrane. Not every single cell on Earth has a cell wall. So don't confuse those two terms. For example, animal cells do not have a cell wall. Plant cells do have a cell wall. I mentioned that in chapter 3 when we talked about polysaccharides, and you needed to know what cellulose was because that was the thing that made up the cell wall of a plant. But again, just to remind you, just point this out, cell walls are different than cell membranes. Everything has a cell membrane. Not everything has a cell wall. Any questions about that? All right. If you're taking notes, you can give your hand a break for this next slide. This kind of talks about, in general, what cell membranes do. And we're going to discuss some of them, and some of them we won't. Cell signaling, we don't really get into that. We talk a little bit about genetics and how we make proteins from DNA. It goes from DNA to RNA to protein, and we'll talk about that later in the semester. But what we don't talk about is that there's reasons this happens, because there's signals that tell your cell, all right, hey, we need this molecule. Hey, we need you to make that molecule. And this is one of the ways it happens, right? There's something called cell signaling. But that's all you need to know about it. And you don't even need to know that for the exam. That's just for your own knowledge. Here we have fibers of extracellular matrix. In this case, they could be, they could be anything. In this case, it might be something that connects this cell up here to this cell down there, right? That could be that, or maybe it has another function. Because here we have something called intercellular joining. So those are just two proteins that are embedded in the cell membrane that are keeping those two cells together. Cell-to-cell recognition. That's important. Like I'm sure you guys know that you need a certain blood when you get a blood transfusion. It has to be the same type because there are cell-to-cell recognition. Because your body would be like, hey, that's not supposed to be in my body, and they'll attack a certain cell, right? Same thing with transplants. Anytime you get an organ transplant, right, you have to have the right type. Otherwise, your body rejects it because of that. Cell-to-cell recognition. Attachment to the cytoskeleton. And if we did chapter 4, you would know what the cytoskeleton is, but it's exactly what it might sound like. Cyto is the prefix for cell. You know what a skeleton is. So the cytoskeleton is basically like the stuff inside of a cell that gives it its shape. And it has to attach to something. So there we go. It attaches to the proteins in the membrane. What's this one? We talked about that. And here's the big one. Here's the one we're concerned with, at least in this chapter. Transport. It's all about transport, right? The only thing we're really focused on this chapter is the fact that cells let some things into their cell, or let some things in and don't let other things out, and some things they need to get out, right? They control what comes in and out, is basically what we're saying. So as far as we're concerned in this chapter, this is the only membrane function that we are worried about, transport. Any questions about that? Okay. Your book calls it an important function, and I put a little star there to remind you, even though I just said it. It is the most important function as far as we're concerned. In this chapter, we're only going to talk about things coming in and out of the cells. So as far as we're concerned, that's the only function we even care about. And again, those other things are obviously important. We wouldn't have life without those things. But for what we're focused on chapter 5, this is the most important. So let's move on. Let's talk about these things. If you download the PowerPoint, you can click this little animation that shows a little bit of stuff, a little bit of transport in action. I don't really recommend it. It's there. You can watch it if you want. I recommend watching the video that I'm going to share with you, hopefully today, but if not today, this weekend. It'll be another one of those quick 12-minute videos, or about 12 minutes, and I really highly recommend watching it. This guy, Hank, does a lot better job of describing what happens than I do because he has better technology and he's more entertaining. So you can see all these things animated. And anyway, watch the video when I post it is what I recommend. And here's yet another video you can watch if you download the PowerPoint. Again, it's an animation. This is more of a realistic animation than the last one, but watch it if you want. Here we go. Now we're getting into it. The bullet points. These smaller bullet points are the last main bullet point. First thing we're going to talk about is passive transport, which you already know about, but I've never used the word passive transport. Then we're going to talk about osmosis, and you know about that. Then we're going to talk about active transport. That should be a new concept to you, at least as far as we're concerned in this class and what the information I've given you. Then we'll talk about something called exocytosis and endocytosis. So here we go. Passive transport is the first thing we're going to talk about. This should be a good review for you because you learned all about this in the lab. First thing you need to know about, and your lab gave it a name, and I don't think your book does, Brownian motion. Molecules vibrate and wander randomly, right? You learned that in the lab. And because of that, we have something called diffusion. Before we even jump into what diffusion is, just look at these pictures down here at the bottom. You don't need to memorize this, but that just shows you the different ways that water molecules can move. And this particular molecule is water. So you can see here, try to imagine like this water molecule, the hydrogen is just pulsating like that. And then on this one, imagine they're pulsating like in opposite ways. And then this one, it's kind of flapping its wings, so to speak. This one, it's spinning this way. This one, Now this one, it's spinning this way. This one, it's spinning this way. And this one, it's spinning that way, I think. Doesn't matter. Those are just examples. The point is that these molecules that you've seen drawn, that you've drawn yourself in lab 2, they don't just sit there. They're constantly moving. Even a one molecule, it's just doing its own thing. Even if it's sitting still in space, right? It's doing those things. Like it's stretching, it's twirling. It might not be going all over the place. It might be sitting there just doing its thing. But because it's sitting there doing its thing, imagine another molecule is right next to it, right here. And then this thing hits it. It'd be like a pool ball, right? And that thing's, it's going to move. And then it's going to hit another one. And it'll hit another one. And it really is. Just imagine like breaking, what do they call that? Racking? When you're playing pool? I'm not from, yeah, racking. So when you hit the balls and they go all over the place. That's essentially what's happening with molecules, right? They're hitting each other. They're bouncing around. And that's what diffusion is. Because of that happening, the molecules move from an area of high concentration to low concentration, right? Until equilibrium is reached. And you need to know that for the exam. That is what diffusion is. And people will mess this up, and it's not trivial. And I'm not big on definitions, but this is an important one, and people will mess it up, and it's not trivial. And I've repeated myself on purpose there. Diffusion is the movement going from high concentration to low concentration, because that doesn't require any input of energy. That's just what naturally happens. Yep, high energy to low energy, or excuse me, high concentration to low concentration. But you're already familiar with this. I'm just probably giving you words to something that you already know happens, especially since you were in lab. You put that little piece of dye on that agar in that plate, right, in that Petri dish, and it was highly concentrated. But then it moved out because the rest of that Petri dish didn't have any dye. So it moved out to the spot that didn't have any. It went from an area of high concentration to low concentration. If I were to set off this tear gas right now, I'm just kidding, I don't have tear gas. But if I were to set off tear gas right now, at first it would really affect me, right? Because that's where it is. But eventually it would move from this area of high concentration to this entire room. This entire room would be filled with tear gas. And that would be the area, that would be the point where equilibrium is reached, right? Once there's an equal amount of tear gas. And I'm surely you could imagine, right? At the very moment that I set off a tear gas canister, it would be horrible right here, and you two would be safe. And then eventually it would get to you guys. And then if there was somebody else in the back, that they would be safe. But then eventually it would get to the point, no matter where you are in this room, it would be equally as bad. Even though I set it off here, it would be equally as bad here as it did there, right? That's diffusion. It would go from an area of high concentration to low concentration until equilibrium is reached. And that's what equilibrium would be. It's like constant, not constant, what's the word I'm looking for? The same concentration, no matter where you are. So yeah. Which one? All right, there we go. So there's your next keyword, equilibrium. Right there, I'll circle it. The next keyword for attendance is equilibrium. So anyway, yeah, that's what diffusion is. You need to know it for the exam. Again, this whole idea of high concentrated, low concentration, it's not trivial because I'm normally not somebody who even cares about definitions, but you need to know how diffusion works because there is something that's basically the opposite of diffusion. And you have to know the difference between the two, not just to understand this chapter, but to understand the whole next two chapters. This is a concept that you need to understand photosynthesis and to understand respiration. You need to understand this idea of diffusion and the opposite of it, which is what we're going to talk about shortly. But anyway, here's a visualization of diffusion. So this is supposed to be, imagine maybe this side right here is the inside of a cell. Here's the inside of a cell. Here's the outside of the cell. This is the cell membrane. The reason it's got holes, that's supposed to visualize that it allows some things in, but not everything, right? That's a semi-permeable membrane. Some things come in, some things can't. So we have a high concentration right here. And imagine, even though you can't see it because it's not animated, these things are bouncing around, right? And if they're bouncing around, eventually they're going to hit each other, and then they'll hit each other, and then it'll keep moving around, just like a bunch of pool balls. And because of that, eventually they're going to keep moving, and then randomly some of them are going to bounce over here, and they're going to bounce over there, and then eventually you'll have equilibrium, right? You'll have the same amount on both sides. Now notice, I love this picture. When equilibrium is reached, the molecules are still moving, right? They're still going left and right, but there's no net movement, right? Meaning the concentration is not going to change. Once you hit equilibrium, the concentration is the same. Yes, some will come this way, some will come that way, but it'll be equal amounts. You hit equilibrium. And again, equilibrium is a very important concept. Equilibrium means that the concentration is both on both sides. In this case, both sides. Of course, when I used the tear gas example, equilibrium was the concentration was the same throughout the room. Here, we're saying the concentration is the same on both sides. So any questions about diffusion? All right. I know this is early, but the next word for attendance is going to be your last name. Now, literally, I mean your last name. So Vance, for example, because I see your name when I'm looking at the screen. When you write the words, your third word will be the word Vance. Anyway, so this next picture, this is the same thing. We're still talking about diffusion, except now instead of looking at one molecule, we're looking at two molecules, the same way you kind of looked at starch and salt at the same time. And here we go, the same thing's happening. It started with the red balls on the right and the purple balls on the left, but eventually, because of Brownian motion, right, because of that random molecular motion, they all moved around until finally equilibrium was reached, at which point the net concentration, the concentration is the same on both sides. So even though molecules are still moving, the concentration doesn't change anymore because you've hit equilibrium. Whew. So any questions so far? And again, I haven't used this word enough, so I want to make sure I'm making this very clear. This is passive transport. So under the category of passive transport, and there's going to be other words, but right now, under the category of passive transport is this diffusion. We've talked about diffusion. Diffusion, which we just got finished talking about, I'm going to abbreviate it here. That is a type of passive transport. And passive transport, it's in the name, right? It's just when things are transported across the membrane, but it does not require any input of energy, right? Diffusion just happens without an input of energy, right? You don't have to heat something up. It'll happen faster if you heat it up, but you don't have to heat it up for it to happen. Anyway, let's move forward if there's no questions about diffusion. All right. So again, passive transport. Make sure you understand this term. First of all, again, diffusion, I call it a subcategory, but it's the diffusion of a substance across a membrane without the input of energy. So in other words, I'm trying to think about how to say this without confusing you. As far as the exam is concerned, it doesn't matter which falls under which. So it doesn't matter if you call diffusion a subcategory of passive transport, or if you call passive transport a subcategory of diffusion. It doesn't matter for the exam. But think about it this way. If we're talking about diffusion, again, the tear gas example I used, that's an example of diffusion. Passive transport, on the other hand, would be more like the picture I showed you, because that's not just any old diffusion. That's when we're talking about diffusion within a cell, right? It's specifically things going across a membrane. And again, the key here for the exam, what you do need to know for the exam, is that when we're talking about passive transport, diffusion is one word you need to associate with it. The other thing you need to associate it with, down the concentration gradient, which is a little bit redundant because you already know by definition, diffusion is going from an area of high concentration to low concentration. So now I'm just saying the same thing. I'm just saying it in a different way. And you should be familiar with me saying it this way too, because I'm going to use these words throughout the semester. Down the concentration gradient. That's the same thing. That means going from an area of high concentration to low concentration. And again, let me underline this too, because it's a very important concept, because you're going to need to know the difference between passive transport and something called active transport, and then we'll get to that later. So right now, that's why I'm emphasizing these things right here. Passive transport, there's no input of energy. And the way you can remember that, it's in the name, passive, right? It's passive. It does not require an input of energy. So any questions about what passive transport is? All right. And again, if you download the PowerPoint, I've got another animation you can watch. Here we go. Let's talk about something else. Substances that do not cross spontaneously, meaning like they won't just happen without some sort of help, is what I mean by spontaneously, or what your book means by spontaneously. Those are transported via proteins. And when that happens, that is called facilitated diffusion. Facilitated diffusion is a subcategory of diffusion, right? So there's all types of diffusion. One of the types is facilitated diffusion. Let me ask you this. All right, it's kind of hard to picture because there's only two of you in here. But imagine this room was filled with 500 people, right? That's a lot of people. Now, if the doors were open, like, all right, there's 500 people in here. I cranked the heat all the way up. We're all hot. We're all uncomfortable. What would happen if I opened the doors and just did nothing? Didn't say anything to anybody. I just opened the doors. What would happen to the people? People start going out, right? That would be diffusion. People would start going from an area of high concentration to low concentration, right? I wouldn't have to input energy. I wouldn't have to push anybody. They would just do it. They'd be glad to get out of here. Now, if the doors were shut and they couldn't open them, they wouldn't leave the room, right? Not because they don't want to. They still want to diffuse from that area of high concentration to low concentration, but they can't make it out. Some things can. You know, that there's a...
Sustained up there.
Yeah, the heat would be even worse around there, yeah.
But then again, we still have like...
That's true. And we wouldn't die necessarily because there's cracks under the door, right? So here we are in this room where people can, with the door shut, people cannot come in and out of this room with the door shut. But gas can. Or if there was a flood, I'm sure water would come down those doors, right? Water would get under the crack. So some things can come in and out of this room. Some things can't. That's the same thing with cells. There's some things that do want to diffuse into or out of a cell because they want to go naturally down the concentration gradient. They do naturally want to go from an area of high concentration to low concentration. But some things cannot make it through the cell membrane. Maybe it's because they're too big. Maybe it's because they have the wrong chemistry. It doesn't matter. The point is they can't make it. So here we go. On this picture, this very left picture, that's what we've already talked about earlier. That's just regular old diffusion. Those little purple balls, they're more highly concentrated outside the cell than they are inside, so they just naturally diffuse right through the membrane and come into the cell. Here we have these things, these triangles and these purple or blue dots. They, again, want to come into the cell because they want to go from an area of high concentration to low concentration, but they can't for whatever reason. Maybe they're too big. Maybe the chemistry's off. Doesn't matter. All right. Hold on. Actually, don't give it to me yet. But, yes, 1 second. Okay. So, yeah, that would be facilitated diffusion. It's still diffusion, except now these things are too big, so they need to be facilitated. And going back to the analogy I was using, the facilitation is opening the door, right? So in this case, there's proteins. So facilitated diffusion, it's still passive transport, right, as we see here. It still does not require energy because, of course, like I said, it's still passive transport. The only thing new here is that we have these transport proteins because whatever thing it is trying to come in and out just can't come in and out without some sort of doorway, so to speak. So, again, you should, when you think about facilitated diffusion, if you're taking like quick notes and like trying to like really abbreviate things, you should put maybe like plus protein, right? It's because facilitated diffusion is the same thing that you've learned about diffusion, except now, good grief, that looks like garbage. Except now there's a protein needed to help these things in and out. So are there any questions about passive transport? All right. Well, 3.5 minutes. All right, I'm going to do the introduction to osmosis, and then we'll finish. You already know this from the exam, but osmosis is the diffusion of water across a selectively permeable membrane. And I like this, and this isn't in your lab. This is not in your lab, and I need to fix that because I think your lab just says diffusion of water across a membrane. But more specifically, it's across a selectively permeable membrane. Or as far as we're concerned for this class, who cares? What we should think of, as far as we're concerned, it's the diffusion of water across a cell membrane, right? And yes, it can happen outside of cells. Obviously, you did it, right? You had those little membranes that were supposed to be like cells, but they weren't actually cells. Anyway, the point is, it's the diffusion of water across a membrane that lets some things in and doesn't let other things in, right? That's what we're talking about. And here's these words I told you in Chapter 3. I told you they'd come back. I said in Chapter 3 you would not need to know them, but you would need to know them later. So here we are. You need to know them. It is now later. You need to know the differences in the relationship between these three words: solute, solvent, solution. And I'll give you an example. When you mix salt in water to make salt water, the salt that you sprinkle in there, that's the solute. That's the stuff that's getting dissolved. The water that you poured it into, that's the solvent. That's the thing doing the dissolving. The salt water that you made, that is the solution. If anybody here knows what simple syrup is, you get some boiling water, you put some sugar in the water, and then you have what's called simple syrup. In this situation, the solute would be the sugar, the boiling water would be the solvent, and the solution would be the simple syrup. I'd go on and on. Just think about it, just dissolving stuff, right? If you dissolve something, the thing you're dissolving is the solute. The liquid you're dissolving it in is the solvent. And the thing you make with that is called the solution. And that is what you guys will need to know. You need to know those words because we're going to be using them a lot. So what was your suggestion for the other word?
Well, spontaneously, but after this.
Yeah, maybe not. Since yeah. Actually, I'm gonna do one more off the wall. And the last word is going to be taco. Like the thing you eat. Taco is the last word. That's up to you. You do some research on that. You go to a Mexican restaurant and eat some tacos. I won't give you any credit for it, but it should be fun research. You could turn, yeah. So the question is, even though, because we have a little bit less than a minute left, what if you write an independent work about tacos? If you can make it related to biology, which you could, right? Because how are you going to grow the beef? How are you going to grow the lettuce? How are you going to make the cheese? Those are all biological processes. But you'd actually have to do some research and talk about it. But anyway, that's it for the class today. I'll be online for office hours at 9 a.m. Make sure you send me the attendance words before 9 a.m. Anything after that, you have to do the questions. Speaking of questions, does anybody have any questions? All right. Hope you guys have a great weekend. And I hope you see me for office hours if you need me. I can help you with your labs and all that stuff.